Hi, my presentation is about on the involvement or role of peroxisome on adrenoleukodystrophy or ALD. Before I start, let me give you some background about the disease before I relate it to the involvement of peroxisome. Adrenoleukodystrophy is an X-linked genetic disorder affecting 1 out of 21,000 people. The disease is caused by a mutation in the ABCD1 gene, which is responsible for the production of ALD protein. Mutation of the gene will result on the production of abnormal or dysfunctional ALD protein. Generally, the disease affects male more than female because of the difference in the number of X chromosomes. Females have 2 X chromosomes, while males have only 1 X chromosome. Even there is a mutation at 1 X chromosome of female, the other X chromosome can still produce functional ALD protein. Male, on the other hand, with only 1 X chromosome present, can be automatically affected in case of mutation. So what will be the effect if there is a dysfunctional or abnormal ALD protein? In the cell, ALD proteins are found to be present at the surface of peroxisomes as transporter proteins. Peroxisome is an important organelle, and one of its functions is the breakdown of very long-chain fatty acids, or the VLCFAs, in the cell. ALD proteins are important structures that are responsible for transporting these VLCFAs inside the peroxisome. Unlike most fatty acids, VLCFAs are too long to be metabolized by the mitochondria, and thus it must be metabolized by the peroxisome. When the body cannot produce functional ALD protein because of the mutation of the ABCD1 gene, very long-chain fatty acids cannot enter the peroxisome. And if these VLCFAs were not able to enter, the peroxisome cannot metabolize these fatty acid chains, so they will accumulate in the body. This accumulation of VLCFAs is very toxic to certain cells. The cells affected include the cells of the blood, cells that produce hormones like the adrenal gland, and the cells that produce myelin like the oligodendrocytes and the Schwann cells of the nervous system. Therefore, clinical signs seen in patients with ALD include those that cause malfunction of the said cells. This X-linked disorder is characterized by the cerebral demyelination and degradation of the adrenal function. Myelin is a fatty white substance that surrounds the axon of some nerve cells, forming an electrically insulating layer. It increases the speed at which impulses propagate along the myelinated fiber. Accumulated fatty acid damages the myelin that surrounds the nerves, thus delaying the propagation of the impulses. This can cause seizures and hyperactivity, and can also cause problems with speaking, listening, and understanding of some verbal instructions. So far, there is no definitive cure for ALD, although treatments like hormone administration, bone marrow transplant, and diet therapy restricting the intake of VLCFAs are considered alternatives. Thank you very much for your attention, and I hope you had learned something from my presentation.